Welcome to this short video in which we explain how you can validate the current direction in your TMS coils yourself. First, why should you measure the current direction in your coils? Although one assumes that the arrows on the coil correspond to the current direction in the coil, we recently discovered that this is not always the case, and that in a small number of coils there is a mismatch between the label and the actual current. When labels are correctly indicated on the coil, an initial anterior to posterior induced current in the brain corresponds to an initial posterior to anterior induced current in the coil, as shown on the left side. The opposite situation is illustrated on the right side. Why do we need to know the current direction? Well, if the current direction is incorrectly labeled, this will cause incorrect interpretation of the study results. For example, within M1, AP or PA directed currents are assumed to probe different neural networks. Here we suggest a simple solution which enables you to measure the current direction of your coils yourself, using a simple paperclip which you can fold into a self-made loop probe. The loop probe is attached to a connector which is attached to a BNC cable. What else do you need? We used a data acquisition unit connected to a TMS system and our loop probe, but you can also use a simple oscilloscope. Next, hold the loop probe on the stimulation surface as illustrated and make sure the signal labeled part of the probe is oriented towards the anterior part of the coil. This figure shows how your signal should look like when holding the loop probe in the center of the coil. The left side of the figure refers to the initial AP directed induced current in the brain, while the right side corresponds to PA. Furthermore, the expected signal of a monophasic pulse is plotted on the top, while the signal corresponding to a biphasic pulse is plotted on the bottom. Here you see how the signal should look like for a monophasic pulse when the goal is to induce an initial PA-directed current in the brain tissue. Next, you see how the signal should look like for a biphasic pulse when the goal is to induce an initial AP-directed current. For more details, we refer to the full publication. We hope you enjoyed this video, and if you are interested in our lab or other work from the research group, go to www.brainslab.com.